Okay, so this is where we left off, where we were just about to begin talking about sex chromosomal abnormalities. These tend to be tolerated more than abnormalities of autosomes. There is no monosomy for any of the autosomes that is compatible with life. Period. None of them are compatible. They are all embryonic lethals. But the X chromosome, it's possible to have this. And the primary reason why is primarily due to this idea of X inactivation. In that, for an individual who has two X chromosomes, and this kind of relies on this idea that you have some concept of X inactivation. So earlier on, did you talk about what X inactivation? I feel like I asked that question already, but you have a basic idea of that. Yeah. So females inactivate one of the two X chromosomes. Is that inactivation complete for that chromosome? No, it is not. There are regions on the telomeric portions of the X chromosome that escape X inactivation. And there's probably about 25 or so genes on the inactive X that are still expressed. Those genes have homologs on the Y chromosome, except for one, which is called exist. We're not going to worry about that one right now. But there's a homolog on the Y. So those genes are expressed on both the X and the Y. And if a female did complete X inactivation, she would have fewer or less gene expression for those 20, 25 genes or so. But the majority of the genes on the X chromosome do get shut off, except for a small percentage. So about 20 genes or so. So incomplete X inactivation is why there's a phenotype. But also the fact that it's a rather small number of genes explains why the abnormalities are not as significant. There are thousands of genes on the X chromosome. So if you were trisomy for the X and you didn't have X inactivation, you would have thousands of genes that are overexpressed. But in a trisomy X, if you have 3 copies of the X chromosome, you actually inactivate 2 of them, as opposed to just 1. So take it down to 1 chromosome. But those 2 inactive X's are still overexpressing a few genes. And if you're a monosomy for the X, you have 25 or so genes where you don't have the proper amount of gene expression. But it's not thousands. So monosomy for the X is Turner syndrome, 45X. And so here's karyotype, 1 X chromosome, no secondary or no second sex chromosome. So characteristics for Turner syndrome, and you certainly want to be able to recognize a description of this. They tend to have short stature. A broad webbed neck is very common in these individuals. They lack the typical growth spurt that you see in adolescence. They oftentimes will have infantile external genitalia as well, even though they should have reached the point of puberty. They oftentimes are very late with respect to puberty. Ovarian dysgenesis, they show streak ovaries oftentimes. They have a triangular shaped face, a lot of kidney abnormalities. Congenital heart defects sometimes are seen. And then when you ask them to stand, oftentimes their forearms angle away from their body for some reason when they stand. So oftentimes you'll see pictures and they're out like this in terms of the picture, and I'll show you some examples of that. 80% of cases, non-disjunction in the father as opposed to the mother. So good question is why? Why more common non-disjunction in males? And rather than belabor the point, I'll give you the answer. It makes me sad, but I'll give it to you. Is what is separating? If we have a female and she's going through meiosis and the X chromosome is synapsing during meiosis, what is it synapsing with? The other X, right? So those 2 chromosomes match up. What about in a male who's going through meiosis? They have an X and a Y, right? Does the X and the Y, do those 2 chromosomes synapse? Yes, they do. So the X and the Y synapse. But the X and the Y have very little in common with each other. Just the telomeric regions of the X and the Y. That's all that synapse. So it's thought that the synapse of the X and the Y is not terribly efficient because it's not along its entire length. And so there can be a greater percentage of meiotic errors that occur with the X and the Y synapsing than what you see with the X and the X synapse. It's about 1 in 5,000 live births, but in terms of conceptions, about 15 to 20% of chromosomal abnormalities that are seen in spontaneous abortions. And so, again, this is a circumstance where Turner syndrome is compatible with live birth and compatible with survival, but exhibits a much higher rate of spontaneous pregnancy loss. So many of these chromosomal abnormalities, they have a high rate of loss. There seems to be a mechanism during embryogenesis to detect when things are not quite right. So here you can see, for instance, the bent forearms in these individuals. This is an individual who is about, I think, 14 years old, and so should be further along in terms of development that she's showing. It's kind of hard to see. This picture is probably the best one in terms of the webbed neck that is oftentimes seen as well, and short stature. And deafness is another one that's commonly seen with respect to Turner syndrome. The next one is Klinefelter syndrome. Klinefelter is 47XXY. So we have an individual who has an extra X, but also has a Y chromosome. These characteristics can oftentimes be subtle. Of all of the different chromosomal abnormalities, this is the one that's most often missed. Probably the majority of Klinefelter males are never diagnosed as being Klinefelter. So they tend to have tall stature with long arms and long legs, normal sexual behavior, poorly developed secondary sexual characteristics. They tend to be sterile, invariably sterile. So oftentimes this diagnosis is made when there's infertility. And then they present to an infertility clinic, and that's when the diagnosis of Klinefelter is actually made. Slightly below average intelligence, but they don't have mental retardation, and small testes as well. Gynecomastia is sometimes seen, and so you can see breast development. They tend to be low in terms of testosterone, and so the amount of body hair tends to be rather low as a result of this, and learning disabilities, but not mental retardation. And then karyotype will show 2 X chromosomes plus a Y chromosome. Individuals with Klinefelter, and you can see how this individual could easily be missed in terms of Klinefelter presentation. Here, this individual showing the significant gynecomastia that's possible. This individual probably very little, but you can see the long arms that are characteristic. This is probably about a 6-month-old who was diagnosed with Klinefelter based on karyotype analysis, but just looking at the child, you would be very hard-pressed to say that you could pick out that that individual had Klinefelter. So this is quite commonly missed in terms of diagnosis. It's pretty close in terms of distribution. 56% derive the extra chromosome from their mother, and so somewhere around 40% or so from the father. So largely a pretty similar mix. Does age play a role? It's unclear at this point. If age plays a significant role in terms of producing a child who has 47 XXY. Trisomy X is 47 XXX, and there's pretty rare to show abnormalities. When they are present, sterility is one. Yep, do you have a question? Okay. So one is Y male, and how does the extra X play a role? And Y male is that any individual who has a Y chromosome and has a gene called SRY, SRY is what initiates the male cascade or pathway in terms of male development. That gene is on the Y chromosome. So if you have a Y chromosome and you have SRY, you will develop male characteristics. Now you can have a X46XX male, and due to a translocation of SRY, actually. So as long as you have SRY, you'll develop male. So it doesn't matter how many X chromosomes you have. You could have 48XXXY, you will still develop male characteristics. Okay. Now, the second is the additional role of the X chromosome. Will Klinefelter's individuals exhibit X inactivation? And the answer is yes, they will. They will inactivate one of these two X chromosomes in every single cell because the mechanisms take it down to one active X. But are there still genes that are expressed on that inactive X? Yes, about 20, 25 genes or so. So what these individuals have is they have about 25 genes that are overexpressed because they're expressed on this X, and this Y. It is a rather subtle phenotype because the number of genes is relatively small, because it's only those that escape X inactivation that are overexpressed. The vast majority of these genes that are on this X are shut down. Does that make sense? Okay. Good. Yep, that's a good question. So, trisomy X, 47XXX. How many X chromosomes will be inactivated in a trisomy X? 2, leaving 1. There will still be overexpression for that set of genes that escapes X inactivation. Sterility, menstrual irregularity, some mental retardation, maybe some learning disability as opposed to mental retardation. 1 in 1,000 to 1 in 2,000 live-born females, so it's pretty common to see. It is possible, and I showed you FISH analysis, where we can have more X chromosomes, 4 or possibly 5 X chromosomes. As you add more X chromosomes, you increase the severity of the clinical presentation. So 4 is worse than 3, and 5 is worse than 4. And especially the mental retardation increases as time goes by. 47XYY syndrome. This would be a male who has 1 X and 2 Y chromosomes. Pretty mild clinical presentation. Men tend to be taller than average, slight reduction in IQ, probably would not qualify as mental retardation. Learning disabilities. Attention deficit disorder is very commonly seen in these individuals. This karyotype was thought, there were papers published in the early 1900s that said that a 47XYY syndrome karyotype predisposed to violent behavior in these individuals. And that notion has been shown to be completely false due to follow-up studies. If you remember the movie Alien 3, terrible movie, hated it. But there was 47XYY syndrome played an important role in that movie. Anybody remember? Remember Ripley? Remember Ripley? Ripley crash landed on a planet that was a penal colony. Anybody see that movie? Oh, jeez. Okay. I think it might be on Netflix. That's your assignment for the weekend anyway, is go watch this movie. But the penal colony was specifically made, the planet was for males who were 47XYY. And I remember when I was sitting there watching this movie, and I think I almost screamed out loud. It's like, no, that is so wrong. That is completely wrong. And yet Hollywood doesn't care that it's wrong. They would have a scientific advisor who could tell them that's not really true, but they do it anyway. And it's so frustrating sometimes. But it's not associated with more violent behavior. Okay. Any question on any of that material? Go ahead, and I will switch to my next set of slides. Yep.
So,
yep.
So with the XYY, how do you get that? Like, what kind of non-disjunction is that, like XYY?
Good question. How could you get that? And I'm debating here about how much of a jerk I want to be. Pretty significant. Now, I'm going to turn it back and say, you show me how you could do this. And I'll give you a hint, non-disjunction during meiosis. But how could non-disjunction during meiosis produce 47XYY? I think if you can do that, you have a good understanding of what's going on. So rather than give you the answer, I'm going to turn it around and say, everybody should do that as an experiment to say, how could you get that? Another good one would be, how could you get 5 X chromosomes? Non-disjunction during meiosis as well. So not trying to be a jerk, but I think it's a good learning experience and activity to try to do that yourself. Okay. Was there a second question as well? Same? Okay. Okay. Genetic basis of infertility and pregnancy loss. So this is going to continue on from here. And this is a talk in previous years that I have not given here. And the talk that I have given here was on gene therapy in this particular spot. And I decided I would do this instead. And then, of course, this year there was huge development with respect to gene therapy. Anybody know what that huge development was in the gene therapy area for humans? A particular therapy that's called CAR-T therapy. Has anybody talked about CAR-T? It's very, very cool. C-A-R space T therapy. Definitely Google that and take a look at it. It is the very first gene therapy approach that has been approved in humans. It costs about $500,000 to have it done. So it's not cheap, but it's very, very cool. And it is with respect to certain forms of leukemia and probably myeloma as well quite soon. But that's a different lecture for a different day. So what we'll talk about here, again, is kind of extending some of these chromosomal things. So we'll be talking about specific abnormalities that affect the ability to conceive, essentially. So this one relates back to what we talked about before, being familiar with all of these terms. One that I want to be sure of is paracentric and pericentric inversion, and the difference between a paracentric and a pericentric. Unbalanced versus balanced rearrangements, and what's called a Robertsonian translocation as well. So those would be the big things that we'll have. Chapter 6, again, the cytogenetics section is good for this. So again, meiosis, playing a really important role here. And one thing I'd recommend is this slide shows the normal process. We've looked at abnormal meiosis, but you really want to make sure you understand normal meiosis as well. Because if you don't understand normal meiosis, you really can't understand non-disjunction occurring. So during meiosis 1 is when our homologs, and notice here they're showing 2 homologous pairs rather than just one in this particular cell. The homologs pair, they synapse, they exchange genetic material, they recombine, they line up, and in meiosis 1, the homologs separate. And that separation in this kind of colorized image shows that there's recombination on some of the arms of these 2 chromosomes as a result of synapse and genetic recombination. Then meiosis 2, when we have meiosis 2, we get then separation of the sister chromatids. So here is the start. We have 2 homologs, separation of homologs. So one of the red homologs here, one of the red homologs here, one blue in here, one blue in there, and then the sister chromatids separate. They separate this way, one goes here, one goes there, and then the blue end is going to do the same thing. So that's normal meiosis. And so you can use that slide to make sure that you understand that basic process. So, abnormalities in structure. Duplications or loss of parts of chromosomes that can occur. As opposed to an entire chromosome being gained or lost, it's just pieces. And we can generally break it down into 2 types. There are balanced rearrangements, and a balanced rearrangement is one that does not produce a gain or loss of genetic material. And then the second one is the unbalanced, where the rearrangement does cause a loss of genetic material. Unbalanced are much more likely to produce a phenotype in the individual where we see it, and that should make perfect sense. Unbalanced, we have lost, or we have gained genetic material. We are going to call it trisomy or monosomy for a particular part of a chromosome, and that can be bad. With a balanced rearrangement, we've simply moved regions around, but not necessarily the lost ones. However, the primary thing we're going to focus on in this particular talk are the balanced translocations. So the next obvious question is, why would we do that? Why focus on balanced rearrangements when they tend not to be associated with phenotype? And the answer is that these balanced translocations oftentimes result in pregnancy loss and reduced fertility. And so an individual who has had multiple conceptions that has resulted in the inability to carry to term, a balanced rearrangement is very possible in one of those individuals. And it must be something that should be analyzed, karyotype, to look for if there's an abnormality there. Many, many times these are so early in development, embryonic lethal, that it's before the woman even realizes that she was ever pregnant in the first place. So a woman may say, no, I've never been pregnant. It's possible that she's been pregnant multiple times, but it's been so early that she didn't even know that that was the case. So inability to conceive or to carry a fetus to term is oftentimes what leads to the discovery that a person has one of these balanced translocations. So, or balanced rearrangements. The most common ones are the translocations. So, again, definition. Interchange of genetic material between non-homologous chromosomes. Chromosome 7 and chromosome 8 are non-homologous. 9 and 22 are non-homologous. There's exchange of material between those. These are the most common ones, and we break them down into two groups: reciprocal translocations and Robertsonian translocations. So we'll talk about both of those. Reciprocal translocations. Probably what causes these are, we saw in meiosis that we have this process of synapse and genetic recombination. Sometimes mistakes are made in that process. So instead of recombination between homologs, we have recombination between non-homologous chromosomes as a mistake. So it's essentially a rare mutation that occurs. So somehow we get breaks on the two chromosomes that are involved. Then those broken chromosomes switch the genetic material between the two of them. And so we produce what is commonly called a derivative chromosome. So a person inherits a reciprocal chromosome or potentially inherits a reciprocal chromosome from one of the parents. So here we have a normal copy of chromosome 3. Here we have a normal copy of chromosome 5, derivative 3, derivative 5. Those two, the derivatives, exchanged genetic material. And the breaks are shown essentially where those arrows are localized. So this portion of this chromosome actually should be on chromosome 5. And this portion of this chromosome should be on chromosome 3. But there's been an exchange. Is this balanced? And typically, yes. This would be a balanced reciprocal translocation. So it will generate 2 DER chromosomes. Another important point is that when you get this kind of a translocation, it involves only one of the 2 homologs that are there. So a person will have a derivative 3 and a derivative 5, but they will also have a normal 3 and a normal 5 as well. This will be really important in the next few slides, that there's also a normal 3 and there's a normal 5. So it's possible that a person could have these derivative chromosomes, and they could pass them on. So it's possible that a person could inherit both of the derivative chromosomes from a parent. So they inherited both the derivative 3 and the derivative 5 from the parent. They probably are not going to have a phenotype because they have a balanced reciprocal translocation. And every single cell will have those derivative chromosomes. And I say this will, I hope, make sense in a minute, because probably at this point it doesn't, because you don't have all the pieces in terms of what's going on. The offspring of this individual are at a high risk for having chromosomal abnormality syndromes or a high rate of pregnancy loss. Duplications, deletions of chromosomal regions. So how is the big question. And I often preface these next few slides as this is some of the most complicated material that I'm going to cover while I'm here. So, interesting time to go. But, so here we have an individual. So here's a parent. okay? And this parent has a normal copy of chromosome 3, a normal copy of chromosome 6, a derivative 3, and a derivative 6. If you look at the colors, you can see that here is black and white. That portion of chromosome 6 is derived from 3. And this portion of this derivative 3 is derived from 6. That's where the translocation has occurred. This parent probably has no phenotype as a result of this translocation, because it's just simply an exchange of genetic material. Is it possible she could have a phenotype? Yeah. If the translocation breaks apart a gene, it's possible that it could cause a phenotype, but oftentimes it doesn't. So the parent has 2 normal copies of these chromosomes, has a normal 3 and a normal 6, and has a derivative 3 and a derivative 6. Maybe she inherited those from her parent, or it was a spontaneous novel mutation. The parent then produces a child with the karyotype that is shown at the bottom. So what is going to happen during meiosis is that the normal 3 and the derivative 6 are going to synapse, and then they will separate. Homologs will separate. And the normal 6 and the derivative 6 are going to separate. That separation is random. So there's 4 different ways that this could occur. A gamete could inherit the normal 3 and the normal 6. A gamete could inherit the normal 3 and the derivative 6. A gamete could inherit the normal 6 and the derivative 3, or a gamete could inherit the derivative 3 and the derivative 6. Because the gamete will always have one of each of these. So if we look at this offspring, one assumption that we're going to make here is that the other parent is normal. So let's say that this has occurred in a male, and they're producing then sperm. And if we look at the chromosomes in this individual, we see a normal 3 and a normal 6. Our assumption is that the other parent is normal. So let's say that that other parent, the one that we're not seeing, donated a normal 3 and a normal 6. So then over here, this parent donated to the sperm the derivative 3 and the normal 6. Could have been 4 possible ways. This is the way that it turned out, where it's the derivative 3 and the normal 6 that came. You can draw this out in greater detail if it's not quite making sense. But if you can see 4 different ways these chromosomes could potentially be presented in this individual, I think you'll get it. So, child inherited the derivative 3. That derivative 3 is here. The tip of that chromosome is derived from chromosome 6. That part right there. This chromosome, this derivative 3, is also missing part of the tip of chromosome 3. And I get a sense that I've probably lost you at this point, but hopefully I'll be able to bring it home as we work through this. So this inheritance of the normal 6, there was a 50-50 chance. This child happened to inherit the normal 6 and the derivative 3. This child is likely to have a phenotype as a result of this. Why? What I've drawn in terms of arrows here is the blue arrows represent chromosome 6 telomeric sequences. So we have one here, and we have one here, and we have one here. So we have 3 regions for the tip of chromosome 6. What about chromosome 3? The very tip, there's one here. Here's the derivative 3, and remember, this is chromosome 6, so the derivative 3 doesn't have the genes for that region. So that particular chromosome is missing the tip of chromosome 3. So the math says that they will be trisomic for a portion of chromosome 6. They will have 3 copies of genes for chromosome 6, and they will be monosomy for portions of the telomeric tip of chromosome 3. So for some genes, they have too many copies, and for other genes, they have too few copies. There is a pretty good chance that this could be associated with a phenotype. Genes tend to increase in terms of density as you move up toward the telomere. So this probably could involve perhaps hundreds of possible genes. The larger is the exchange, the greater is the phenotype, most likely. So translocations can be very highly variable between individuals, exactly where the breakpoints occur. So the chromosomes are involved, the breakpoints that are involved, highly variable. So it's almost impossible to predict what the phenotype is going to look like because each translocation is unique. But the idea that it could cause a malformation in the resulting offspring or could potentially cause lethality, it should be able to make sense. And you should be able to estimate the risk as well of this, of how this occurs. And the larger the translocations, the greater is the phenotype going to be. So one more thing that I want to do here is this is showing one example in terms of the malformation that could occur. So there's really 4 different conceptions that are possible here. One is that this parent who has this translocation could donate a normal 3 and a normal 6 to that particular germ cell. So, first important point, could this individual produce an offspring that has a perfectly normal genotype and phenotype? Yes. What is the likelihood? It would essentially be 1 in 4, 25% chance. Is that valuable, important information? Absolutely, yes. You know, you have this balanced translocation. However, there's still a 1 in 4 chance that you could produce a perfectly normal conception. Now, can we do things like in vitro fertilization and then pre-implantation genetic diagnosis to test for which chromosomes have been inherited? Yes, we could do things like that. It is also possible that you can inherit the derivative 3 and the derivative 6 together. That's a balanced translocation. Genotypically, it's abnormal, but phenotypically, it's probably normal. If the parent was normal, then the offspring is normal. So in terms of phenotypically normal conceptions, there's about a 50% chance that it's normal. Genotypically, there's a 1 in 4 chance that it's normal. Then you can have unbalanced. In this case, derivative 3 and normal 6, but it's also to have normal 3 and derivative 6 as well. Would those two produce different phenotypes? Probably it would, because you're dealing with trisomy and monosomy, but flipped the other way around. So those two could produce abnormalities, possibly lethal conceptions, and maybe slightly different phenotype between the two. That may not make full sense. You may have to work on that a little bit in order to understand it. And I said earlier, this is complicated, and it's hard to understand. But work it through. Diagram it in terms of how it's happening and how you can produce the 4 different conceptions. If you can understand that, you should be good. Any question on that before I move on? Okay. But you can certainly stop by and ask me about it. We can diagram it as well. Robertsonian. Robertsonian translocations are quite interesting. These translocations involve only the acrocentric chromosomes. This is why earlier I said probably knowing which ones are the acrocentric chromosomes has value to you. 13, 14, 15, 21, 22. Those are the acrocentric chromosomes. The translocations that are here involve loss of the short arms of 2 of the acrocentric chromosomes and fusion of the long arms at the centromere. So in this diagram, we have, here's a chromosome 13, here's a breakpoint, here's a chromosome 14 and a breakpoint. The 2 long arms, the Q arms, fuse together to produce a new metacentric chromosome. And usually there's also this really small chromosome that consists of just acrocentric, and that's typically lost from the cell. So you wouldn't see that chromosome present because oftentimes it lacks a centromere or functional centromere and just kind of goes away. So breakpoints, fusion, producing this, producing a second fragment that usually you're not going to see. This is a common mistake that I've seen in many textbooks, where they talk about human chromosome 2, and there's very strong evidence that human chromosome 2 is a result of a fusion of 2 primate chromosomes. And actually, if you look at chimps, they now designate chromosome in the chimp as 2p and 2q, because they correspond to the p arm and the q arm of human chromosome 2. And oftentimes, this is called a Robertsonian translocation that occurred, but it's not. I remember I was going through one of my genetics textbooks that I have, not the one I recommend you guys read, and they called this a Robertsonian translocation. So I looked up the email address of the author, and I sent him a nice little note and said, you know, I think you made a mistake here. And he was very cool about it. He said, yeah, I think you're right. And so then the new edition came out, and so the first thing I did was quickly go to that section to see if I could find if he had changed it. And he did, which was really neat to see. Another cool aspect of this is that there's a paper that described this, and the sequencing of this region, you find telomeric sequences at this site in human chromosome 2. That is a remnant of the actual fusion between those 2 chromosomes. And so that also shows you that it's not a Robertsonian translocation, because a Robertsonian translocation wouldn't result in telomeric sequences at that site. So it's an aside, but it's interesting. So the acrocentric chromosomes, 13, 14, 15, 21, 22. The genes in these acrocentric regions are the ribosomal RNA genes, if you recall. These are oftentimes called balanced translocations. They're truly not balanced translocations, but they are close to a balanced translocation. So these individuals tend to be phenotypically normal who have one of these translocations. So we have fused 13 and 14 together, or at least 2 of those versions, to produce this new chromosome. Have we lost genetic material? Yes, we have. So there's 5 chromosomes that are acrocentrics. We have 2 copies of each of those acrocentrics. So in a diploid cell, there's going to be 10 chromosomes that have ribosomal RNA genes. If we have a person who has this translocation, they've lost 2 of those. So they express ribosomal RNA genes at 80% of normal. Is 80% of normal for ribosomal RNA expression acceptable? And yes, it is. So that translocation by itself doesn't produce a phenotype, because we still have 80% of what we normally would have. It's not balanced, but it's as close as you're going to get to balance. They have 1 fewer chromosome. So these individuals will be 45X, or XX, or 45XY. So if you get a board question and you see that it is 45 chromosomes, and you also see that they have 2 sex chromosomes, so it's not Turner syndrome, then it is very likely to be a Robertsonian translocation. It has produced an individual with 45 chromosomes instead of 46. So, what's the big deal? And the big deal is offspring. Offspring are the ones that suffer from these reciprocal Robertsonian translocations. And how? We see duplications and deletions in the potential conceptions. And I skipped over this one in terms of karyotype nomenclature, but I'll come back to it now. 45XY, derivative 1314, Q10, Q10. That is a typical designation for Robertsonian translocation. And this is weird knowing which ones are the acrocentrics. So if you see 45 chromosomes derivative and the 2 chromosomes are both acrocentrics, you're golden in terms of knowing that that's a Robertsonian translocation. So how does this happen? Second most complicated thing that I talk about whilst I am here. So both in the last 10 minutes or so. We have 45 chromosomes. This is, and in this particular Robertsonian translocation, it's involved chromosome 14 and chromosome 21 instead of 13, 14. Both acrocentric still. We have a parent who has this translocation, the fusion to produce a new metacentric chromosome. they still have one copy of chromosome 14 and one copy of chromosome 21 present in their cells because they're diploid cells. Right? They should normally have 2 copies of 14, 2 copies of chromosome 21. They have 114, 121, and 1 fusion chromosome. They will go through meiosis. What happens during meiosis? Chromosome synapse. What's going to happen during synapse? And what will happen is that these 3 chromosomes will synapse together, as opposed to 2 chromosomes synapsing together. It's a very unusual arrangement. So in this particular segment designated A, this is the 1421 Robertsonian translocation chromosome. Okay. This is chromosome 14 that is synapsed here, and this is chromosome 21 that is synapsed here to this region. Right? The blue regions will synapse, and the pink regions will synapse together. So that's what we're going to see. Now, what happens during meiosis is the chromosomes separate. When you have 2 chromosomes, one goes one way, one goes the other way. What's going to happen when you have 3 chromosomes as opposed to 2? We're still going to get separation. But there's 3 different ways that the chromosomes could separate. So now things are getting complicated. But essentially, we're going to see separation. But which way? 3 different ways. So the first one, and I've tried to color code this, what we're looking at here is what's called alternate balanced, and you don't have to worry about that nomenclature. Essentially, this is showing the germ cells that would result with this separation. So we have one germ cell that has just the new metacentric chromosome. That means that this chromosome has essentially separated up and to the left. That means that these 2 chromosomes are going to go down and to the right. With 3 chromosomes, it's always going to be that 2 go one way and 1 go the other way, unless you have non-disjunction, and let's not even worry about that right now. We're assuming that disjunction is okay. So this is one possibility. Up to the left and down to the right produces a cell that has these 2 chromosomes, in other words, these 2, plus a cell, a gamete, that has that one. So that's the first one. Then the second one is where these 2 chromosomes go together. The one in quadrant A and quadrant B go together, and the one in quadrant C goes by itself. So that will produce this set of germ cells. Chromosome 14 by itself, and then this one, which is the metacentric, plus chromosome 21. So that's the second one. And then the third one is the last of the possibilities. And that is where chromosome 14 goes down and to the left, and these 2 chromosomes go together and up to the right to produce that germ cell. And that will lead to this group. Here's one cell that has chromosome 21 by itself, and then this one that has chromosome 1421 translocation plus chromosome 14. So what that means is 1, 2, 3, 4, 5, 6 different germ cells. Combinations could be produced by this individual who has a Robertsonian translocation. The next step, these are the germ cells. The next step is fertilization. So let's assume this is occurring in a male. So the Robertsonian translocation is in the male. They're producing 6 different types of sperm. Those sperm can fertilize what we're assuming is a normal egg. What does the normal egg have? The normal egg will have 1 copy of chromosome 14 and 1 copy of chromosome 21. So the next step is to essentially do that with each of these different germ cells, the 6 up above. So if we start with the first one, this germ cell, is a 1421. So putting those together, everything is normal. So that will produce a perfectly normal conception, both genotypically and phenotypically. This one. If you bring this chromosome along with these 2, you essentially reproduce the same pattern as we saw in the parent, where that individual will have, and they call it balanced here, balanced in quotation marks, a balanced reciprocal translocation that was the same as the parent. Genotypically abnormal, phenotypically most likely normal. The next one. Here we have chromosome 21 plus the metacentric chromosome, bring those together. The first one that we will see is that we will have 2 copies of chromosome 21. That's there. We will have this copy of 14. That's there. And we will have the metacentric chromosome here. Is that problematic? We have 2 copies of chromosome 14 material. We have 3 copies of chromosome 21 material. This would essentially be a translocation Down syndrome offspring or conception. Trisomy 21. The next one is this one, chromosome 14 by itself. If that sperm fertilizes a normal egg, you have 2 copies of 14, you have 1 copy of 21, monosomy for 21, that would be lethal. So that conception would not survive. The next one, here, we have the translocation chromosome plus chromosome 14 fertilized, or this sperm fertilizing a normal egg, then we'll have this combination. Where here's 21, we have a second 21 there on that chromosome. So here's the 21 from the egg, the 21 from the sperm. Those are okay. But we get 1, 2, 3 copies of chromosome 14, trisomy 14, and that would be lethal as well. So that would be a lethal conception. And then the last one is chromosome 21 by itself. And hopefully we're starting to get it now. Bring these together. We have 2 copies of chromosome 21, only 1 copy of chromosome 14, monosomy for 14, also lethal event. And so we have 6 possible conceptions, 1, 2, 3, 4, 5, 6. 3 of them are lethal conceptions because they're monosomy for autosomes. Those will go away. So in terms of live conceptions, there's about a 1 in 3 chance that it's a translocation Down syndrome. And actually that number in terms of live offspring is lower than that. It's more like a 1 in 10 because of the higher rate of pregnancy loss with Down syndrome. But at least amongst conceptions that could survive longer than early lethalities, they're about a 1 in 3. A 1 in 3 chance of reproducing the pattern in the parent, and about a 1 in 3 chance of having a normal karyotype. So, parent comes in, they have done genetic analysis, they know they have a Robertsonian translocation. They sit down with you and say, what is the likelihood that we could have a normal offspring? You could sit down with them and work through this. Maybe not today, but after you study it a little bit more, that you would be able to do that. That's complicated stuff, no doubt about it. But is it important? Absolutely it is. One aside, this is kind of an interesting, how much time have I got? A minute. I'll give you an aside and then I'll stop for today. Is I wrote a board question many years ago, so you're not going to see this question. And I asked this. And my question then was the likelihood of seeing, I think, a translocation Down syndrome amongst all conceptions. So the correct answer is 1 in 6. Is the likelihood amongst all conceptions that it could be a translocation Down syndrome. Eventually, I saw the question. I was reviewing a board booklet that had the question in it, and somebody in their wisdom changed it to offspring and kept it at 1 in 6 as the correct answer. Is that true? No, it's not. I was so mad. Ugh. Couldn't believe that somebody changed the question that way. Because amongst offspring, take that one out, take the monosomies all out. And also, you have a higher rate of pregnancy loss with a trisomy 21. So it is not 1 in 6. It's more like probably like a 1 in 10 chance. The last thing I'll say, Robertsonian translocations are responsible for about 5% of all Down syndrome cases. The risk of Down syndrome reoccurrence is normally considered to be really, really low. The likelihood it could happen again. But if it's due to a Robertsonian translocation, the likelihood of recurrence is very high. It's the same as it was with the first conception, maybe about a 1 in 10 chance. So if there's a Down syndrome conception that occurs, it is a really, really good idea to do chromosomal analysis of both parents. Rule out Robertsonian translocation as a cause. And once you rule out Robertsonian translocation, then you can give them a good estimate based on their age, the likelihood that it could happen again. But you could see, let's say you had a 22-year-old female who has a Robertsonian translocation and you don't know it. What are you going to say in terms of the likelihood of it happening again? Probably like 1 in 1,500 is the likelihood. What's the real likelihood if she has a Robertsonian translocation? 1 in 10. Very different numbers. So identifying this is a really good idea. So if you have a patient who has a Down syndrome conception or a Down syndrome birth, chromosomal analysis looking for Robertsonian translocation is a good idea. Okay, I'm out of time. Any questions? How many slides did I have left? Okay. So what I will do is I will continue this tomorrow and wrap this up quickly, and then we'll move into genetic screening. And I think we should be able to finish on time. Okay, thanks very much.